Myself Pallavi Sharma. We are going to discuss some important question about the chapter Biomolecules. So we'll start with first question. Which one of the following graph shows the relationship between the rate of an enzymatic activity and substrate concentration? So as we have learned the topic enzyme are biological catalyst, and what their function is to increase the rate of reaction. Since they are increasing the rate of reaction, so the activity of enzyme increase with if we are making that velocity, this is representing enzymatic activity. This is the substrate concentration. If we make graph of this, so with increase substrate concentration, the enzymatic activity increases to a certain point. When the substrate concentration reaches to the maximum, it reaches to the V max. Here it is V max. So the correct answer is D. Next question, moving on to the next question. Which of the following statement about enzymes are correct? So enzymes do not alter the overall change in free enzyme energy for reaction. This statement is correct because enzymes affect the rate of reaction, but they do not alter the free energy of the reaction. Second is enzymes are the protein. Yes, enzymes are 3-degree or tertiary proteins which have active site. Next is enzymes speed up the rate of reaction by lowering the activation energy. So yes, we have studied this graph where we have studied that rate of with introduction of enzyme, the activation energy reduces. This is the activation energy with enzyme. This is activation energy without enzyme. So activation energy reduces. Next is enzyme speed up the reaction by lowering activation energy is correct. Enzyme are highly specific. This is also correct. And enzyme input needed to start a chemical reaction. It is known as activation energy. So activation energy is actually what is the activation energy is minimum energy required to carry out or initiate a chemical reaction. So the answer is D, all of these. Next question is, it is said that elemental composition of organisms that are inanimate object, like inanimate means non-living object on the earth crust, are similar in the sense that all the major elements present in both, like carbon, hydrogen, about these. Then what will be the difference between these two? Option 1 is living organism have more gold. Living organism do not, or they have do not, or very minute quantity of gold is present in living body. Second is living organism have more water. Even though we are comparing with non-living, but still we can't differentiate on the basis of water because earth's surface has more water. Next, living organism have more carbon, oxygen. This is the right answer because living organism possess percentage of more carbon, hydrogen, oxygen and other substances in their body or their mass than inanimate object. That is why C is the correct answer of this question. Okay, next question is enzyme that catalyze the removal of group from substrate by mechanism other than hydrolysis. So first of all, we are discussing what is the function of ligase. Ligase is responsible for bond formation. Liases, they are responsible for breakdown by oxidation. Hydrolases, as the name suggests, they are responsible for hydrolysis. They are also responsible for breakdown, but they are responsible for hydrolysis, that means breakdown in presence of water. Then next is dehydrogenases. These dehydrogenases remove hydrogen atom and substitute on next other atom. So the correct answer will be liases B. Next question. Read the following statement and select the correct option. Statement 1. Ribozymes are the RNA molecule which catalyze the synthesis of certain specific RNAs and removal of introns from mRNA. So this statement is correct because ribozymes, we have studied that a ribosome contain like 23 srRNA or 28 srRNA. These are peptidyl transferases, and all of these are ribozyme. Okay, so these ribozymes are RNA particles which are responsible for catalyzing a chemical reaction, but these are not proteins. These are made up of RNA particles. So statement 1 is correct. These are proteinaceous. So we have studied above that these are RNAs in nature. So second statement is wrong. So answer is C. Statement 1 is correct. Statement 2 is incorrect. Moving on to the next questions. Next, enzyme catalyze the biochemical reaction by activation energy. So yes, they catalyze the biochemical reaction by activation energy lowering. We have studied that what is activation energy. Activation energy is minimum amount of energy is required to initiate a chemical reaction. So enzymes are the catalyze biochemical reaction by lowering the activation energy. Next is another question regarding the enzyme kinetics. So here they have asked the question about x-axis and y-axis. So we have to analyze this. See, this is the graph they have given. We have to find out what is given in x-axis, what is given in y-axis. See, if x-axis is the rate of reaction and y-axis is enzymatic activity, so if the rate of reaction increases, enzymatic activity do not increases at all. We have studied earlier. Second is if the x-axis is enzyme, vice versa, so it will not affect the rate of reaction at all. Next is enzymatic activity at x-axis and pH. See, enzymes are tertiary or 3-degree structures of protein. If we increases the temperature, what will happen? That hydrogen bond will break down, that will cause denaturation of protein. So increase in temperature will affect or reduces the enzymatic activity. So this can't be the correct answer. The D, with increase in that pH or temperature, enzymatic activity which reaches to a certain point, that is Vmax. So D can be the right answer. D is the correct answer. Next question. Read the given statement and select the correct option. Right end of a polysaccharide chain is called as reducing, while the left end. See, in a polysaccharide chain, there are two ends, right end and left end. The right end is responsible or very active, while left end is non-reactive. So we can mention it, very active end is known as reducing end, non-reactive end is known as non-reducing end. So left end is non-reducing. Starch can hold iodine molecule. Yes, this is correct. We have studied this earlier when we have mentioned polysaccharide, that starch has ability to hold iodine molecule in its helical structure, but cellulose do not possess this activity. So this statement is also correct. Next is starch and glycogen are the branched molecule. Yes, starch contain amylose. Starch is a homopolymer of glucose, and glycogen is also homopolymer of glucose. So they are the branched molecule. While fourth statement is starch and glycogen are the reserve food material. This statement is correct both in animals and in plants respectively. Both is correct. So all the statements are correct. Correct answer is D. Moving on to the next question. Refer the two reaction 1 and 2 and given below and select the correct option. See, first reaction ADP plus PI, this is a formation reaction. And during a formation reaction, we have to remember that energy is utilized. Okay, and this is a breakdown reaction. And during a breakdown reaction, energy evolve. So the energy utilized reaction is known as endergonic, while energy evolve is known as exergonic. Now match the correct answer, which is A. Next question is inhibition of succinate dehydrogenase by malonate. So we have studied the competitive inhibition when a substrate unable to bind with, or when a enzyme is unable to bind with substrate but it bind with its competitive inhibitor, So this will prevent further reaction. So this is known as competitive inhibition. The correct answer is B, competitive inhibition. Next question is dihydroxyacetone 3-phosphate and glyceraldehyde 3-phosphate are interconvertible. See, this is a reaction of glycolysis. This is the fifth reaction of glycolysis, where DHAP is formed from fructose 1,3-bisphosphate breakdown, and DHAP, which is 3-carbon compound, is formed, and along with that phosphate glyceraldehyde 3-phosphoglyceraldehyde structure is formed, that is DHAP and PGAL. This is also a 3-carbon compound, and these both are interconvertible with each other. A reaction which is catalyzed by an enzyme which cause interconversion of these molecules is the enzyme known as isomerase. So the correct answer for this question is A. Moving on to the next question. Whole enzyme. Whole enzyme is a complete enzyme, consists of an apoenzyme and cofactor. Up till now is the correct thing. Select the option which correctly identify the nature of that apoenzyme. We have to remember that apoenzyme is a protein part of an enzyme, while if we are talking about that coenzyme or cofactor, these are non-protein part. So the correct answer is A, protein and non-protein. Next question. Inhibitor which closely resembles the substrate in a molecular structure and inhibit the enzymatic activity by binding the activate site in the enzyme is known as, the same answer is the known as competitive inhibitor. Okay, we have studied the this question earlier, the same question earlier, so the answer is C. Next is this maltose. So we have studied that maltose is a oligosaccharide, and this oligosaccharide have 2 monosaccharide unit, which is glucose plus glucose. So they have asked about breakdown of enzyme, which enzyme is used to break this reaction. So in this reaction specifically, this is a hydrolysis reaction because we are using H2O molecule. So the correct answer is hydrolysis, because we are using water molecule for the breakdown of maltose molecule to produce 2 glucose molecule. Next is what is denoted by X and Y. Here see what we have studied, that when we apply enzyme to a biological or a chemical reaction, it reduces its activation energy. So in X we can consider that X is the activation energy with enzyme, while Y can be, it is higher than X, So this is activation energy without enzyme. Since we have find out that what is x and what is y, then let's find out the option correct. Activation energy without enzyme, so x is not, a is not the correct answer. Substrate concentration, they are not talking about substrate concentration at all, so b is the most probably correct answer. This is the correct answer, b. Let's move to the next question. Next is cellulose is a homopolymer of glucose, which is correct. Yes, it is a homopolymer. Inulin is a homopolymer of fructose. This statement is also correct. Next is starch gives blue color and glycogen gives red color. This statement is not correct because glycogen gives brick red or reddish brown, you can say this as brick red, reddish brown color to iodine solution. So because of this, third statement is wrong. Fourth statement is cellulose gives no color to the iodine solution. Yes, because of its branching, it is unable to bind with iodine solution. So the correct answer is A. Statement 1, 2 and 4 is correct. Moving on to the next question. Read the statement and select the option that correctly identifies X and Y. In glycogen molecule, successive glucose unit joined together by X and branches are linked with Y. So when we have studied the glycogen structure, we have read that there is a branching present and a linear chain is also present. In case of a linear chain, there is formation of 1,4-alpha glycosidic linkage. So in glycogen linear chain, 1,4-alpha glycosidic linkage. While if we are talking about branching, so in branching there is same glycosidic linkage but it's with 6th carbon, so 1,6 glycosidic linkage. So the correct answer is B, 1,4 glycosidic linkage. Moving on to the next question. What does A represent? See, we have studied the structure of adenosine triphosphate. This is ATP. Okay, this is the first carbon, second, third, fourth and fifth. The pentose sugar is bound with one side to the nitrogenous base and to the other side to the phosphoric acid. This is a nucleotide structure and this bond is known as glycosidic, while this bond which they are asking, this is known as phosphodiester bond. So the correct answer is B, that in this question what they have asked, that what A is representing. So A is representing the phosphate bond. Next question. The inhibitor which does not resemble to the substrate and bind to the enzyme at the site other than the active site. So this is the question which is explaining that it is not, the inhibitor is not binding to the substrate site. The substrate is binding to the exact site. Like here is the substrate, it is bounded with enzyme, but there is additional information. What they have mentioned here, that inhibitor which does not resemble to the substrate in structure and bind to the enzyme other than the active site. This is already the active site, so they do not bind with the active site, they bind with the at this position. Like at this position they are binding. This site is known as inhibitor site, and such structures are known as non-competitive inhibitor, because they are not competing with substrate for the binding, they are finding their own position for the binding at the enzyme surface. Moving on to the next question. Feedback inhibition of an enzyme. So see, if we are making A product, A is making B product, B is making C product, C is making D product, and D is making E product. If E product is too much formed, it will give negative feedback to D. Due to this, D will give negative feedback to C, C will give negative feedback to B, and B will give negative feedback to A. So what will happen? Feedback inhibition of the enzyme is influenced by the end product, because E is the end product. The activity or the concentration of E is describing that the reaction has to be stopped. So at this time E is giving the negative feedback. So next question is same as this is Michaelis-Menten equation. This is about, so which one of the given graph shows the effect of pH on velocity on a typical enzymatic reaction? So the correct answer about this is C, because see, here is enzymatic activity and here is pH on x-axis. If we are increasing pH, the enzymatic activity is increasing to a certain point. After it reaching to the neutral pH, enzyme start showing their reduce in their or starting to the max of the activity, and after it reaches to the neutral pH, the activity of the enzyme reduces to a certain content. So the correct answer is C. Next is Michaelis-Menten constant. This question number 22. So Michaelis-Menten constant is V is equal to V max. This is substrate concentration divided by Km plus substrate concentration at initial. Km is Michaelis-Menten constant, and Km depends upon substrate concentration. Substrate concentration which attain rate of reaction at max velocity. So the correct answer is C. So the correct answer is D instead of C, because in C it is written half. So the correct answer is D. Moving on to the next question. Here they have asked about what of the following option correctly identify structural formulae. So first we are going to discuss about this structure. See, this is first carbon, second carbon, third, fourth, fifth and sixth. So this is a hexose 6 carbon compound. So 6 carbon compound here is fructose is also a 6 carbon compound. Glucose can be, but we have studied fructose is never formed the 6 ringed structure. So A option is not correct. B and C can be the correct. D is also correct according to A structure. Now moving on to the second. This is first carbon, second carbon, third carbon, fourth carbon, fifth carbon. So this is definitely a 5 carbon. So this can't be the D option because fructose is a 6 carbon. This is wrong. Ribose, this can be ribose. And we have studied that A can't be, A is not the correct answer. So C is the correct answer because they have mentioned that second position and third position both are occupying OH group. That is why C is the correct answer. Next option, next question. Big four element. Big four element. Big four is representing the major element which compose the living system, and these are the most essential element of the living system. So these are carbon, hydrogen, oxygen and nitrogen. So according to this, A is the correct answer. Moving on to the next question. See, select the option correctly identify the chemical bond. First structure is polysaccharide. So each polysaccharide contain glycosidic linkage. Moving on to the next, protein, they have peptide linkage. Moving on to the next, fats, they have ester bonds. And water, it is bounded by hydrogen bond. I don't think so I have to explain the structure of glycosidic linkage. So correct answer will be glycosidic, peptide, ester and hydrogen. B is the correct answer. Moving on to the next question. In a DNA molecule, phosphate group is attached at 5' end of the sugar residue of its own nucleotide and 3' end of the sugar residue of the next. So अगर हम इसको स्ट्रक्चर के अकॉर्डिंग देखें, so see, this is a deoxyribose, HH, second carbon, OH, H, here H, and this here is nitrogenous base is attached. Here CH2, and this group is going to attach with phosphate and this phosphate oxygen is going to bind with third carbon. This is third carbon. So this bond is known as phosphodiester bond. Okay, so what they have asked, 5', this is fifth carbon and this is third carbon. So 5 and 3 is also completed and the bond between them is known as phosphodiester linkage. So A is the correct answer. Next, moving on to the next question. Match the column 1 with column 2 and select the correct. So see, cotton fiber Cotton fiber is made up of cellulose. So A with fifth option. Then exoskeleton of cockroach, we have studied that exoskeleton of insect is made up of chitin. So this is chitin. B is going to meet with third. C, liver. Liver is present in human body and its main function is to store glycogen. So C is with second option. Next is peeled potato. When we have completed the plastid, we have studied that potato tuber is responsible for the storage of starch. So D is going to match with first. Last, roots of dahlia. So we have studied that root of dahlia and artichoke are rich in inulin, which is fructose polymer and also present in bacteria. So E option is going to match with fourth. So 5, 3, 2, 1, 4. So correct answer is A. Next question is, give the structural formula is correctly identifying along with the related function which of the following option. Okay, so this is a structure. See, this is first carbon, second carbon, third carbon. This is RCOOH, one fatty acid. This one is second fatty acid, and this is COC, that is ROR. This is representing ester bond. This is also an ester bond. So if something is representing ester bond, that means we are talking about fat molecule. So what we have studied, in lecithin there is presence of two fatty acid molecule attached to glycerol molecule, and at the one end it is attached to the phosphate compound. So see, this at one end, third carbon is attached to the phosphate compound. So this is B is the correct answer. Lecithin, a compound or component of the cell membrane. Moving on to the next question. Next is identify X and Y. See, when we are studying about a amino acid chain, it contains amine group at 1, carbon alpha at second. Here is CONH group, which is peptide linkage. Then again carbon alpha, which is C double bond OH, H and R2. So if we are talking about 2 carbon polypeptide chain, this end is known as amine end, which is represented by N. This is known as carboxyl end, which is represented by C. So in a polypeptide chain there are 2 ends are present. One is known as N terminal, known as amine terminal. Second is known as C terminal, which is known as carboxylic acid terminal. So answer is A. Next question. We have studied one practical or in our book that read the given statement and select the option that correctly sort these with respect to A and B in a given flow chart. We have taken the living tissue, which is grinded in a trichloroacetic acid, because Cl3 and acetic acid, trichloroacetic acid. We have made thick slurry. Then this thick slurry is stained with cheesecloth and 2 fractions are obtained. One is acid soluble pool and another one is retentate or acid insoluble fraction or pellet. Okay, so what will be obtained in these two? We have to categorize them. One is molecular weight ranging from 18 to 800 Dalton, that means bio, micro, molecules. We are talking about biomicromolecules. Okay, so if these are biomicromolecules, so these will be found in first acid soluble pool, second protein, nucleic acid and polysaccharide, and lipid. These all are biomacromolecule. If something is biomacromolecule, these will be found in B. We will be writing B in front of it. Third, it contain element that have molecular weight more than 800 Dalton. Anything that have weight more than 800 Dalton will be categorized as biomacromolecule. So this is also present in B. It has monomer. So monomer will be present in acid soluble pool. So it will be categorized as A. Generally has polymer. So B already has polymers. We have already learned this. So let's categorize this. First and fourth both are present in acid soluble pool. So A is 1, 4. Okay, and B contain second, third and fifth. Second, third and fifth. So C is the correct answer. Next, moving on to the next question. The correct order of chemical composition of living. See, in a living tissue, the most important thing which is present in them is water. Moving on to that, protein is maximum. Then apart from them, carbohydrate are maximum. Apart from that, nucleic acid, that is DNA and RNA, and then lipid, because all the membrane are made up of lipid and 6 chromosome. Then iron, because signal transduction is important. So correct answer is C. Moving on to the next question. Which of the bond indicated by X and Y? See, this is a X bond. This is 5th carbon and this is 3rd carbon. And we just said that bond between 5th and 3rd carbon, PO4 group is present. This X bond will be phosphodiester bond. If we talk about Y, that cytosine and guanine, these are bounded with each other with 3 bonds, and these 3 bonds are hydrogen bond. So X is phosphodiester, Y is hydrogen bond. What will be the correct answer? B can be the correct answer. Okay, moving on to the next question. Galactose, this is a fact. Galactose is present more often or mostly present in our nerve cell or brain cell. So this is also known as brain sugar. So categorizing galactose as a brain sugar. 3, anticoagulant. Heparin is an anticoagulant. Fructose, it is a fruit sugar. So this lecithin, we have just studied that lecithin is present in our membrane. Second, then insulin is a protein or a hormone. So it is a kind of protein. So 1, 3, 4, 5, 2, 1. D is the correct answer for this question. Moving on to the next question. 3 structural formulae A, B, C are given. Here identify them and select the correct option. C. 1. This representing adenine, because NH2 group is present at the terminal. This is representing adenine. If we are talking about this, adenine is bounded to the pentose sugar, but here no phosphoric acid is bind. So this is a nucleoside. Because we have studied that nucleoside is pentose sugar bound with nitrogenous base. So this is the pentose sugar bound with adenine nitrogenous base, this nucleoside. Okay, so B option is a nucleoside. Third, a nucleotide, because we have studied that when a pentose sugar bound with nitrogenous base plus phosphoric acid H3PO4, this can be single or double, this depends, that structure is known as nucleotide. So we have identified B and C as well. A is adenine, second is nucleoside, so first option is incorrect, second is adenine, nucleotide, B is correct option. Okay, moving on to the next question. Triglyceride are the fatty acid ester of glycerol which are formed by esterification of fatty acid, 3 fatty acid with 1 molecule of glycerol. See structure here. See, this is the structure of triglyceride. These all three R1, R2, R3 are representing fatty acid, and this is the structure of glycerol. So this is a triglyceride, true fat. Here it contains 3 fatty acid and 1 molecule of glucose. So correct answer is 3(1)C. Next is last question for the video. Reducing sugar. See, reducing sugar are those which can reduce Cu2 positive to Cu positive. Cu2 positive to Cu. This question is based on Benedict solution. The Benedict solution contain cuprate ion. These sugar, what these sugar do, these convert cuprate ion into cupric ions. So these are the conformation reaction. The sugar give them to the red, brick red or orange color to the Benedict solution. So all of these are reducing sugar apart from this sucrose. This sucrose is a non-reducing sugar. So the correct answer is A. This is all about this video. These are all the questions. Hope you have got the correct answer. If you have any doubt, do let me know. And thank you so much.